Sterility testing for injectables. This is a type of quality control testings which is being carried out on the pharmaceutical products, particularly in case of fermentation industries. So there are several tests which comes under quality control tests that we have already studied in theory in the last semester. So one of the quality control testing is to check whether the pharmaceutical product is or any product which is to be used in or for human consumption like injectables, whether the products are free from any type of microorganisms or not. So that is carried out by using sterility testing. So we have a background already, we have studied this, so we'll learn this very briefly in the introductory section. So the aim of this practical is to perform the sterility test of pharmaceutical products. So we know the basics of sterilization, the definition of sterilization. So this sterility test, it assesses whether the sterilized pharmaceutical product is free from contaminating microorganisms or not. So this sterility test procedure, it is designed according to the pharmacopoeia, particularly British pharmacopoeia. In India, we have Indian pharmacopoeia. So these are the protocols which must be followed according to the guidelines given by respect, respective pharmacopoeia. The significance of this performing this sterility test is very clear. It is the additional check and continued compliance with the test does give the confidence to the efficacy of sterilization or aseptic process. For this, we make use of several media. So here we make use of different media so that we can assess whether the fermentation product or the formulation is free from microorganisms such as bacteria and fungi, particularly aerobic and anaerobic microorganisms. So we make use of different nutritional media that will allow the growth of aerobes as well as anaerobes. For allowing the growth of anaerobic bacteria, we make use of thioglycolate broth. So thioglycolate broth is a nutritionally rich medium that will allow the growth of anaerobic bacteria. Now the thing is that thioglycolate broth is a semi-solid medium which is used in sterility testing in order to allow or in order to check the growth of anaerobic bacteria. But thioglycolate broth, it is a multi-purpose enriched differential medium which is used to determine the oxygen requirements of microorganisms. This means that this thioglycolate broth, it supports the growth of all the types of microbes which have differences in their oxygen requirement. So depending on the differences of oxygen requirements, there are several categories, like some bacteria are anaerobic, some bacteria are facultative, some bacteria are aerotolerant, some bacteria are strictly aerobic. So these are the various categories, and these are based on the classification is based on the requirements of oxygen by particular microorganisms. So it is an enrichment broth which is most frequently used in diagnostic microbiology or bacteriology. So this broth supports the growth of anaerobes, aerobes, microaerophilic and fastidious microorganisms. Now the principle of thioglycolate broth. The composition of thioglycolate broth is it is nutritionally rich. It contains glucose, that is dextrose, then pancreatic digest of casein, which is a rich source for nitrogenous substances and carbon substances. Micronutrients are provided by yeast extract. Then there is presence of this L-cysteine, which has a specific function, and all these are the different types of growth factors which are present in this medium. Now, sodium thioglycolate. Sodium thioglycolate is a principal component which is present in the medium, and this is responsible for the consumption of oxygen. It is a reducing agent, and it consumes oxygen and thereby allowing the generation of anaerobic conditions. It is a semi-solid medium. So due to the presence of sodium thioglycolate, which is responsible for the generation of anaerobic conditions, it allows the growth of strict anaerobes also, for example, Clostridia and several other strict anaerobic bacteria. So even the medium is exposed to oxygen, the external oxygen, environmental oxygen, still the growth of strictly anaerobic bacteria it can be obtained due to the presence of the reducing agents such as sodium thioglycolate, also L-cysteine. So this sodium thioglycolate it acts as a reducing agent and neutralizes, and also it neutralizes the toxic effects of the heavy metals if they are present in the pharmaceutical product, like preservatives. So mercurial preservatives. So the toxic effect of mercurial preservative it is neutralized. It also neutralizes the peroxides which are formed by microorganisms in the broth. Thereby, it promotes the anaerobiosis and it allows or it promotes the growth of anaerobic bacteria. So, all these favorable conditions are generated by sodium thioglycolate as well as by L-cysteine, and it makes the medium more favorable for the testing of materials which contains heavy metals. So sodium thioglycolate is a reducing agent which maintains a low constant oxygen concentration level by removing molecular oxygen from the environment. So this is how we can see the growth of or the behavior of bacteria on the basis of their oxygen requirements. So here you can see uh, what is the behavior of aerobic bacteria. So aerobic bacteria, so wherever the oxygen is available despite of the presence of sodium thioglycolate, so oxygen will diffuse in the upper layer, environmental oxygen. So here this is a semi-solid medium, so we can see that oxygen will diffuse from this direction, and the concentration of oxygen will decrease, the diffusion will decrease as we go in this direction. So there will be formation of a gradient, high concentration zone and zero concentration zone. So this is called as a gradients according to the requirement of bacteria for oxygen. The bacteria will grow in that particular zone. So strictly anaerobic bacteria will find at the top because the oxygen concentration is higher here. So this will be the pattern for the growth of obligate aerobes. Then this is the zone of fluid thioglycolate broth where the oxygen concentration is very less or nil. So that will be the favorable area for the growth of anaerobic bacteria. So this will be the pattern for obligate anaerobes. Facultative anaerobes will grow in this pattern. So they will grow in oxygen in almost all concentration zones. So this will be the pattern for facultatively anaerobic bacteria. Aerotolerant bacteria, aerotolerant anaerobes also will be able to grow at the top because they can tolerate the high concentration of oxygen. So this will be a more diffusible form. This will be a concentrated form at the top, and later on it will be a diffusing pattern, but much difference can be seen uh, between these two tubes of facultative anaerobes and aerotolerant anaerobes. This is how we can identify the growth requirements for oxygen for different types of bacteria. So this is how the growth will appear. You can see the first tube here. The strictly anaerobic bacteria are growing in this bottom zone. Then this is the pattern for facultatively anaerobic bacteria. These bacteria have a limited oxygen requirement, so they are called as microaerophilic. So they grow under low oxygen tension. So this will be the pattern for microaerophilic bacteria. For example, lactic acid bacteria, they have this kind of pattern. This is for Clostridium. You may find the examples for facultative uh, bacteria as well. Then the strictly aerobic bacteria will grow only at the top, and at the bottom there will be absence of growth. This is how practically we can see the growth of different types of bacteria having different oxygen requirements in fluid thioglycolate medium. So aerobes and anaerobes, they can be identified by growing them in test tubes of thioglycolate broth. So, so this pattern we have already learned in the last slide. So there will be the different patterns of growth for aerobes, anaerobes, obligate bacteria, facultative bacteria, and microaerophilic bacteria. Now the requirements, other requirements for this practical will be to allow the growth of aerobes also, including bacteria and fungi. So there is use of nutritionally rich medium for this. So nutritionally rich medium is sterile soybean casein digest medium that will also allow the growth of fastidious microorganisms which have special growth requirements. So this is nutritionally rich medium, and which is known to support the growth of almost every uh, bacterial contaminant. So this will favor the growth of aerobes, particularly for aerobic bacteria. Sterile fluid thioglycolate medium tubes, as I said, this will be useful for the detecting the growth of anaerobic bacteria. But practically talking, principally talking, this medium is favorable, or it will allow the growth of almost every kind of bacteria with differences in oxygen requirement. So that is uh, in case of differential, that will be the purpose of differential is used as differential medium. But here in sterility test we'll be using this for the purpose of detection of anaerobic contaminants present in pharmaceutical product. Then sterile Sabrod's broad tube for the detection of fungal contaminants, and the pharmaceutical product to be tested. We have already learned that if it is powdered one, then we have to make use of different diluting fluids. So for depending on the uh, physicochemical characteristics of the product, whether it is an ointment, whether it is a liquid, whether it is a powder, whether it is rich in fats or lipids, accordingly we make use of polar and non-polar diluting fluids. So this part we have already discussed in theory. You can go back and check the notes under the title of quality control testing, that is sterility testing. So the procedure is very simple. There are two techniques. If the sample is available in large quantity, in large volume, and if it is a flowable liquid, then there can be use of membrane filtration technique. And another technique where it requires very less amount of sample, it is direct inoculation technique. So all these techniques we already learned in last semester. I want you to go back and read those notes as well. So here we'll be using direct inoculation technique, and the product that we'll be using is water for injection, that is WFI. So water for injection is used, or it comes with the powdered antibiotic vial. So where this water for injection, it is taken in syringe and it is then injected into the vial of antibiotic where the powder is present, and then the suspension is prepared, and then that suspension is injected. So we'll be using that water for injection as a sample. So that sample is directly inoculated into the sterile uh, nutrient tubes separately, like soybean casein digest medium. It is inoculated into fluid thioglycolate medium, and it is inoculated into Sabrat's broth tube. Then these tubes will be, so by mistake agar is written here. So all these tubes are incubated for to allow the growth of aerobes, anaerobes, and fungi for 48 hours at 35 to 37 degrees Celsius, and then following 48 hours these tubes are observed for the growth. So the observations and results. So if there is no growth in any of the tubes, so here I have written this observation that no growth was observed in any of these tubes. So you can make a table, like in column number 1 you can write the name of the medium, column number 2 name of the medium, column number 3 name of the medium, and below that you can write the your observation, presence of growth, absence of growth. So you can write absence of growth in all the tubes, and therefore uh, our conclusion will be the product under the test passes the sterility test. So this is how we write the conclusion, observation and results.